Okay, so let's get started. This is in continuation. In fact, next 2, 3 classes also would be telling you about the cloning vectors. It's just that they will be different cloning vectors. Some will be plant vectors, some will be mammalian vectors, some will be expression vectors. So just be ready for the various types of cloning vectors, because that is what would be required when you do recombinant DNA technology, the selection of cloning vectors, which is very, very important depending on the need, your specific need. Okay. So so in my last class I had covered the plasmid cloning vectors, and today's class is on the phage cloning vectors. And when I talk about the bacteriophages, the two phages that I'll be covering would be the lambda phage and the M13 phage. So this is the whole battery of vectors I'll be discussing. We are done with the plasmid vectors, lambda vectors, and up till cloning vectors, I think we'll be able to cover today. And tomorrow we'll cover this part of the lecture. So when we talk about the lambda phage, I guess you remember I had mentioned that lambda phages, they can have a lytic or a lysogenic life cycle. So just a brief overview of the same. In case of lytic life cycle, the lambda phage, it injects its DNA into the bacteria, which replicates, makes more of its copies. And once it makes more of its copies, simultaneously the DNA, which is coding for the viral coat protein or the tail protein, or let's say the various proteins which are involved in the uh, lambda phage, they are produced within the bacteria. They use bacterial host machinery to generate both for replication of phage DNA as well as production of the head of the phage particle or the tail. And once these are made, they get assembled into the new phage particle, and they make multiple phage particles are made in the range of 100 or thousands, and finally they lyse the host cell, and this progeny then infects other bacterial cell which are present in the vicinity, and so the whole thing, the chain goes on. I'll just tell you that when you have to, harvest these phage particles, how do you set up the experiment and how do you visualize that where are the phage particles? Okay. So this is lytic life cycle, and coming to the lysogenic life cycle, it is that lambda phage, it injects its DNA into the bacteria, it circularizes, and then it gets integrated into the host genome, and then it stays there as a lysogen, so, or as a prophage. So this we had discussed when I told you about integration and excision and all these things in the gateway vectors I had mentioned last time. And but yes, this is when the phage DNA gets incorporated into the bacterial chromosome. We say that it is maintaining a lysogenic life cycle, and at some point when the conditions are not apt, and let's say stress related conditions, there is a possibility that the lambda DNA gets excised out and enters into the lytic life cycle and just releases itself from the bacteria, which is in a way, let's say, under a lot of stress condition and is dying because of nutrient deprivation, etc. So yes, these are the two life cycles. Then coming to that, how do you set up the experiment? So on my left are the bacterial colonies. Let's say we want to we inject the means we transform the bacteria with a plasmid which has an antibiotic resistance. So how do you see for the bacteria which have taken up the plasmid is in the form of bacterial colonies only. So what you do is that you take these bacteria after transformation and you plate it on a LB agar plate. So the bacteria which have not taken up the plasmids, they die, right? While the bacteria which have taken up the plasmid, they are seen as beautiful colonies. And if, let's say, I grow the bacteria after transformation on a LB agar plate which does not have antibiotic, how will my plate look like? It will be a complete lawn of bacteria. because then I have not given any selection. So I will see a complete lawn. But if the bacteria has taken up a plasmid in which antibiotic resistance is there and I grow in presence of antibiotic, I see separate colonies. So how do I visualize when I am performing, let's say, this lysis of bacteria by these lambda phages? So what I do is that I take my bacteria, I incubate it with the phage. The phage will then attach to maybe a subset of bacteria which are present in my tube, and then I plate that mixture on a LB agar plate. Okay, so what will happen that the bacteria in which the phage had not attached, they will grow on this LB agar plate, and that we can see as a lawn, or let's say turbid thing as a background. But the bacteria which had taken up the lambda phage, it will undergo lytic life cycle, and because it will undergo lytic life cycle, so that bacteria will give rise to progeny of lambda phage particles. Those let's say hundreds of particles that are released, so they will infect another bacterial cell which is present in the vicinity, and so on and so on and so on. And finally what we see is, let's say if I incubate this plate for a long time at 37 degree, I will see that the size of these plaques, so these, where is the cursor, yeah, so these are called as single, or let's say plaque forming units, and the more I keep it at 37 degree and incubate it for longer time, the larger will be the plaque size. And now let's say if I want to harvest these phage particles, so I can, and means in certain conditions, you are you only want to harvest a particular type of phage particle, then you can use a cut tip and take out this section, and then dissolve it in a buffer so that phage particles are in a liquid. And in which condition am I saying that you want to select let's say a specific plaque only, is in those cases, for example, when I'm doing genomic library screening and I come to know that my probe of interest, my gene of interest is in this particular region, after I perform a technique called southern hybridization, etc. So then in those cases, I can harvest a particular this plaque. So till now, what I've done is that I've just introduced you to the fact that there are certain bacteriophages. These bacteriophages, they have the capability to inject their DNA into bacteria, and through lytic process, there is a possibility that the DNA that has been injected, it makes multiple copies of itself, and then means it makes more of phage particles. So this observation was used by various molecular biologists, and the idea came that if we include our gene of interest or our DNA of interest into the phage genome, then obviously multiple copies will be made, just like they are made in case of plasmids. So even this methodology using phage vectors can also be used for cloning purposes. And one thing that I want to emphasize is that the infection process is much more efficient as compared to the transformation process, just for your information. Now coming to the details about the lambda phage DNA. So it is double stranded and it is 49 kb in length. At the ends of it, we see single strand stretches of around 12 base pair each, and these both, they are complementary to each other. Because these both are complementary to each other, they come together and form these cohesive sites. So they are also called as cohesive sites, and they can complement with each other and then make a circular molecule. So this is a general scheme for lambda phage DNA. Then when it makes more of its copies, so by rolling circle mechanism, it shoots out multiple replicated copies, and they are they are in the form of multimer. and each of the individual phage genome, it is attached with the help of COS sites. So means this is one phage genome, let's say 49 kb, this is another 49 kb, this is 49 kb, and so on. So these are, they are also called as concatemers or catenanes, various names are given. So this is how the replication takes place. Now, once this replication is taking place inside the bacteria, and we want the phage particles to be reassembled, then what we have to do, we have to cleave this DNA. So how this cleavage takes place at individual COS sites, because they are the ends of the phage genome, it is with the help of a terminase complex, and that terminase complex consists of genes called gene A endonuclease and neuron proteins. Okay, so neuron and A proteins, these are the proteins which are part of the terminase complex, and their job is to recognize the COS sites, and only when they recognize the COS sites, the DNA is released as a single unit, and this, when this DNA is released, then it gets incorporated into the pre-assembled lambda heads. So just want to say that this is a separate process taking place, but at the same time, pre-assembled lambda head is the lambda head is pre-assembled into the bacteria, and similarly the tail also. So when these two are separately present, they cannot just combine together. It's only when the DNA gets incorporated within the pre-head that we see that the tail attaches. So this is how the final assembly of the this lambda phage particle takes place, and then they are released from the bacteria after lysing. Now, coming to that, how do we incorporate our gene of interest or section of DNA into this lambda genome? So, if you remember that means our aim is to incorporate DNA, preferably for larger size. For smaller size DNA, plasmids are good enough and they are very easy to work with. But if we want to incorporate larger DNA fragments, then we prefer lambda phage. And these phage cloning vectors, they have the capacity of around 20 kb, depending on which phage vector we are talking about and which type of phage vector we are talking about. Now, the problem with using the lambda phage genome as such for cloning purposes is the fact that it has some problem, or let's say some limit for packaging of the size of the DNA within the phage head. If you see that the genome has to be incorporated into the phage head, and it was seen that if it is smaller than certain size, let's say 37 kb, or it is more than 52 kb, then it will not be incorporated into the phage head. And if it is not incorporated into the phage head, then obviously we will not have multiplication, or let's say more phage particles being produced, and the whole purpose of cloning will be defeated. So that's what packaging efficiency is the problem, and 78% to 105% of the lambda genome only can be incorporated. Beyond those limits, it cannot be. Okay. So one problem was this, if we wanted to use a lambda phage genome as the cloning vector. Another problem was the fact that because this whole genome, it's 49 kb, and the some restriction enzymes that we were interested in, there were multiple sites of those throughout the genome. And if you remember our multiple cloning site that we had introduced into the plasmid vector, our aim is to have unique restriction sites, because if we want to clone something, let's say in the eco-R1 site in the lambda phage genome, then because there are multiple eco-R1 sites, we will fragment the vector itself, which is never desired. So these were the two problems that needed to be tackled before using phages as the cloning vector. So how did they tackle these problems? So they carefully studied the phage genome, And what they realized that there are certain regions in the phage genome that are not required if we want to use this lambda phage for cloning purposes. Because in this case, so there was, let's say, some region was non-essential, it was not required neither for lambda replication nor for its packaging, etc. Okay, but apart from that, there was another region which was only required if the lambda phage is going for lysogenic life cycle. So which are those regions? They will be the regions in which are involved in either integration or excision process. So it was realized that how about we delete this whole region. So it is around, let's say, 10 kb region. So it means that now my cloning capacity of the lambda phage, if I remove the non-essential part, would be around 10 kb. 10 or let's say 13 kb, because ultimately 52 kb can be cloned uh, inside, or let's say packaged inside a phage genome head, phage head. So one way to tackle the problem was this, and another problem was the presence of multiple sites for a particular enzyme. So how did they finally get the lambda phage genome which has only one, let's say, equar1 site? So if it was the current times, then the best way to do it is by site directed mutagenesis, and there'll be a separate class for the same, as well as I think in assignment also there is a lecture on the same. But that time when they were actually developing these as the cloning vectors, that was not that technique was not there. So what they went for was for this natural selection process. So what they did was that lambda phage, this is its genome, they infected infected the bacterial cell, in this case E. coli, with this lambda phage. So what will happen, because they infected the bacteria which had equar1, so they selected a bacteria which codes for equar1. So they selected that bacteria. So what will happen, that when this phage will inject its DNA into the bacterial cell, most of the phage DNA, it will be degraded. If you remember that restriction and methylation system which is present as a way of immunity to foreign DNA by any bacteria. Okay, but there is a possibility that as a natural selection process, some of the equar1 sites, they get mutated. If some of the, let's say, one or two equar1 site, they got mutated in a particular phage, uh, this DNA, then, and there is a chance even that, let's say, equar1 didn't act upon the rest of the ones, so you will get a phage particle, or you will see a plaque, some random plaque in the plate in which you had plated this mixture, in which the phage DNA had been mutated, and it was safe from the attack by the equar1. And when you now you use this as the parental strand, or let's say parental phage, and then again go for the same process. So there is a possibility that rest of the equar1 sites are also lost in some of the natural selection process for survival. So by this way, they finally could get a phage particle in which, let's say, there was only single equar1 site. So coming to these phage vectors, I've told you that 49 kb was the original lambda genome, of which around 10 kb has been removed, and these were used as insertion vectors. And there is a single unique restriction enzyme site here. And as I told you before also, their cloning capacity is around 10 kb. Okay. Then they realized that 10 kb is just similar to the plasmid vectors, and is there spatial use of these vectors? So is there a possibility to increase the cloning capacity of these lambda vectors? So then they came up with the idea of replacement vectors. Okay, so let me just first go through one slide on lambda insertion vectors, in which non-essential region has been removed, and The insertion vector itself has become around 35 to 40 kb, and that's why you can clone around 10 kb extra in these vectors. So there are two lambda insertion vectors which are commercially available. In fact, much more than those. Two examples are given here. And what they have done is, because they also wanted to find a strategy for selection of the recombinant clones. If you remember, in case of plasmids, for example, we studied about the blue-white screening. So similarly, they wanted to have one option in the lambda vectors also. So what they did was that they incorporated the LAGZ gene inside this let's say multiple cloning site that they have introduced in ecoRNA. Okay, so let me first explain this. So for example, they had this one ecoRNA site that was left. In that ecoRNA site, they incorporated a multiple cloning site here. So, and that multiple cloning site, near to that, they also cloned this LAGZ gene. So, if LAGZ will be disrupted by the multiple cloning site, if your gene of interest has been cloned in it, then we will get a clear colony. Otherwise, we'll get a blue plaque. That's the right word when I'm talking about phages. That in one case, I'll get a blue plaque. In one case, I'll get a clear plaque. So that's how I can select for the recombinants. And another way what they did was that they introduced at the MCS or ecoRNA site this CI gene. So CI gene is a gene which promotes lysogeny. So the non-recombinants, they will form a turbid plaque because the lytic life cycle will not be there, but some bit lysis has been promoted. Means I'm saying it promotes lysogeny, but it's not that no lysis is there. So what you see is, let's say, turbid plaques, but not clear plaques. But if your gene of interest has been cloned here, then what will happen, that your CI gene, it will be disrupted. And like I told, insertional inactivation, that's the term that I used last time. And because this will be disrupted, so what we will get would be clear plaques for the recombinants and the turbid plaques for the non-recombinants. So this is how these lambda insertion vectors have been developed, and this is the story about those. Now coming to the replacement vectors. So here what they did was that they removed further region of this lambda genome, again which was not much essential. And then they maintained it by adding a stuffer fragment, which is nothing but some random fragment, just to maintain the vector inside bacteria. Because if, let's say, I had removed this whole fragment and now my phage DNA is much less than 37 kb, then you can understand that this DNA will not be incorporated into the phage head. Rather, it will be lost. So just to maintain it, there is a possibility that you add some random stuffer fragment here. So these are lambda EMBL4 series of lambda replacement vectors that they have developed. And at the end of the stuffer fragment, what they have added are the 3 restriction enzyme sites in these orientation, which is given here. And there is a now possibility that you take out this stuffer fragment, you you digest it with any of these enzymes, and you have your gene of interest, which is also cut with the same enzymes, and then you ligate it, and then you have these lambda replacement vectors that are developed. So the major difference between insertion and replacement vectors is the fact that these can incorporate around 20 to 23 kb, and their limit is only around 10 kb. So let me tell you that if you want to construct a genomic library, which is, for example, for human DNA in a bacteriophage lambda vector, then what will be the steps that will be required? So what you will do is that you will cut your DNA, the organism that you are interested in to make the genomic library. You will cut it with a restriction enzyme. In this case, a 4-base pair cutter, SAW3A1 has been used. So what you will get that your DNA has been randomly digested into around 20 kb fragments. This is your bacteriophage lambda DNA, actually the replacement vector. So this is the stuffer fragment. So the stuffer fragment, let's say it's in between BAMH1 or one of the side which is on either side. So I cut it with BAMH1 and what I get are the two ends and this is removed. Now I ligate these ends with this. Now if you see that I have cut this DNA with SAW3A1 and this with BAMH1 with the idea that because these two are compatible ends, they both make GATC as the compatible ends, so the ligation will be very easy between the two. So one question could be that why I did not cut my the organism, in this case human DNA, with BAMH1 itself, because then also it will be compatible ends and I will get my gene of interest ligated into the vector. So generally when you make DNA for genomic library, you want random cuts throughout the genome. If the bigger is the recognition site, then the number of sites throughout the genome will be comparatively less. So what rather we go for is a shorter recognition site. If it is, let's say, for a 4-base pair cutter, you can imagine its presence will be much more prevalent throughout the genome as compared to if I use, let's say, 6 or 8-base pair cutter. So that's why for more randomness in cutting the DNA throughout the genome, we use a 4-base pair cutter. And in this, smartly what I've done is I've cut it with I've chosen a 4-base pair cutter which makes compatible ends with one of the enzyme here, BAMH1, which was present on either side of the stuffer fragment. So once I have this vector ready and this DNA ready, I ligate it, and you can imagine that different random fragments, they get incorporated inside these vector DNA ends. Once I have this ligated DNA, I can package it into the phage head. So how that method is done is with the help of a technique called as in vitro packaging, because we have to find a way to put this DNA inside a phage head. So this in vitro packaging step I will discuss shortly when I'll be telling you about cosmets. I'll revisit that. So again, I've told you that about the phage vectors, and I've already told you of the two methods of selection of the recombinant phages, or let's say screening of recombinant phages. One is by using LAGZ and one is by using CI gene. And I've told you if you use a CI gene, or for insertional inactivation, the recombinants will be in the form of clear plaques and the non-recombinants will give turbid plaques, and similarly in the case of LAGZ based selection method. Now there is third method that is also used in some cases, which is selection using SPI phenotype. So let me just tell you this method of selection as well. So the full form of SPI is sensitive to P2 phage, uh, sensitive to P2 phage. Okay, so it means, so let's talk about the bacteria. So we are talking about a bacteria in which within the bacterial genome there is a P2 phage which is staying as a prophage. It means that let's say we infected this bacteria with a P2 phage that injected its DNA inside this bacteria, and it got integrated into the bacterial genome, and that's why I called it as a P2 prophage. Now this bacteria which has this P2 prophage, it is resistant to the infection by lambda phage. So if I infect this bacteria with lambda phage, it will not be able to, get its DNA inside the bacteria. So why it cannot get its DNA inside the bacteria is because it is sensitive to the product of a P2 phage. So this P2 phage, it for example generates a protein SPI, which is present on bacterial this membrane, because of which the lambda phage is not able to attach itself to this bacteria, and then it cannot inject its DNA. Okay, so that is what is the case in case of wild type lambda phage. So what they have done is that the genes which are actually sensitive to the product of the P2 phage, those genes names are red and gam genes. Okay, so all these notes are present in the note section. You can go through the links or the notes for the same. And here what they have done is that in the phage vector, they have put the stuffer fragment in the region where red gam gene is there. Okay, so this stuffer fragment has the red and the gam gene. That's what I want to say. So if the stuffer fragment is removed and we have the recombinant being made, so it means that the recombinant phage DNA will not have the red and gam gene. If the red and gam gene is not there in the phage genome, so as a result, it will be able to easily infect this bacteria and make more of its copies. And this is how a selection can be made for the recombinant phages as compared to the non-recombinant phage. Okay. Now coming to the need for vectors with larger inserts. So what we have discussed till now are the plasmids for it, which had like 10 kb insert cloning capacity. Then we had these bacteriophages which have around 10 to 20 kb of cloning capacity depending on which vector we are using. But it was very soon realized that when we are talking about genomic libraries, and that too of organisms which have very large genome, then in those cases we would want to have the vectors which have much larger cloning capacity. So when we are talking about those vectors which have very large cloning capacity, then we have to generate those vectors. So I'll just come to those list of vectors that have been generated. So we have already talked about these three vectors. Now, there are other types of vectors that have been developed by the molecular biologists so that they have higher cloning capacity, like 30 to 44 kb for cosmid vectors. Then there is phage vectors. There is P1 bacteriophage, which has, so it was later, discovered that like lambda phage, there is another bacteriophage called P1, in which the size of insert that can be cloned is around 70 to 100 kb. So then there are some vectors which are modification of P1 phage. They have like 130 to 150 kb cloning capacity. Then we have BACs and YACs which have cloning capacity much higher. And as you can see for YAC, which is yeast artificial chromosome, the cloning capacity is around 2 MB. So some of these we'll be discussing tomorrow, but just for your information, because at least for the genomic library, we need very large insert size, for the ease of maintenance of the library, and also because if we want to screen a genomic library, if we have too many clones to be screened, it will become extremely difficult to find your gene of interest or your clone of interest. So for that purposes, it will be best if your genome of the organism you are interested in is present in a genomic library which has limited clones rather than, let's say, several thousands or millions of clones. So let's understand that how, or let's say that the formula for selection of a vector, and this formula is telling us that how many, n stands for the necessary number of recombinants. Let's say how many recombinants need to be screened to have, let's say, 99% probability to find your gene of interest. Okay, so I hope that you understand what the formula is, that this formula is telling us that how many recombinant need to be screened to have 99% probability to find your gene of interest. So here n is necessary number of recombinants. P is obviously we can't keep, let's say, 100% ever. So we keep it as 99%. And F means the fraction of the genome. And in this case, what is fraction of the genome? Is the insert size. So fraction of the genome is insert size. Let's say if it is for plasmid vectors, then I'll keep it as 10 kb. If it is for lambda replacement vectors, I'll keep it, let's say, around 23 kb. And which organism we are talking about, that is G stands for the whole genome size. So this calculation has been done for this human genome, which has around 3 billion base pairs. So the number of clones that need to be screened if I make my human genomic library in a phage insertion vector, phage replacement vector, which has insert capacity around 20 kb, then I have to screen 688,000 clones to find my gene of interest. So you can understand how difficult it will be for me to, maintain these clones in the bacteria, and also maintain, I mean, in the lab, because these are like huge amount of number of clones to be maintained. And also the screening will be extremely tedious. You have to plate these many number of clones. On the number of plates, you use very big plates for screening purposes, and then only you can find your gene of interest. So for all these purposes, it's important that we have cloning vectors with bigger cloning capacity. So in this case, this context, let's get started with one artificial vector that was made, which is Cosmet, in which very smartly, they have increased the cloning capacity from, let's say, 20 kb that was known till that time to up to 48 kb, or let's say 40 kb. So what they have done in Cosmets is, so let's say it's a marriage, or let's say a cross between plasmids and phage. So in this case, what they have done is that they have taken a plasmid backbone, plasmid vector backbone that I told you in my last class, which will have three major components for sure. One will be obviously the origin of replication, because without this, the DNA cannot replicate. One will be a selection gene, which is, for example, in this case, ampicillin is written. And of course, there should be a multiple cloning site. So these are some basic plasmid backbone thing that are present in the Cosmet. What one thing extra is present in these vectors is the cos site. Okay, so if you remember, cos sites were the ones which were responsible for entry of the phage vector into the phage head. Without the cos site, the DNA cannot enter the phage head. And in this case, we are fooling the phages in the context that they don't care what is present inside. Okay, just because the cos sites is present, so anything that is present inside can actually enter the phage head, and then we can maintain it. So again, going through the points, it is cross between phage and plasmid. It's a circular DNA, and the cos sites are present, which will be helpful and utilized for packaging inside the phage heads. And they have considerably higher capacity of cloning as compared to plasmid or phage alone. So how will you go about cloning in these vectors? Let's go through that. So we generate our genomic DNA fragments of around 35 to 45 kb. And this is my Cosmet vector. And I cut my Cosmet vector at the polylinker site, or let's say also called MCS, multiple cloning site. with the same enzymes which are present at the ends of these genomic DNA fragments. Once I cut these both with that, then I perform ligation reaction. But I perform ligation reaction in such a way that multimers are being produced. How do I ensure that? So a lot of research was done for that. So if I have this if I have these fragments, the inserts in very high ratio as compared to the vector, then generally what we see means they have actually standardized the conditions by which concatemers are made, which are all joined by the cos site. Okay, so now we have ligation reactions, and from here to here, what I have at the ends, I do have this plasmid backbone or cosmid backbone. At the ends, I have cos site, and in between are my big DNA fragments that I cloned. Okay, now I want to get these into the phage head. So what I do is that I incubate it with the lysate, or let's say by with a protein mix which has everything for a generation of phage head and the tail. How do we do this? So again, this process is called in vitro packaging, but because at each end we have a cos site, so one whole genomic phage equivalent DNA will get incorporated in one phage head. Okay, none of the phage head can obviously accommodate two of these. So because of these cos sites, we have somewhere pooled the phage head to incorporate this whole DNA which didn't have any phage gene except the cos site. Okay, once these recombinant cosmate virions are made by in vitro packaging, how in vitro packaging is done, I'll again come to that. Then you infect your E. coli bacterial cell with this phage. Because it is a complete phage particle, it will inject its DNA inside. Once it injects its DNA inside, then it can stay here as a plasmid. How it will be able to stay here and maintain itself as a plasmid is due to the fact because it has origin of replication. Okay, so you can look for your recombinant bacteria which have taken up the inserts by maybe ampicillin resistance, or maybe in some of the cosmid vectors they also have this lag Z based selection, or some other selection is there. Okay, so this is how the whole cosmid thing is done. Now I will discuss about the in vitro packaging in my next class, and then I'll start on with the other vectors that are there for cloning. So I'm open for questions.